Howdy, my name's Phil, and you're probably wondering what the hell this video is about. Well, buckle up, ladies and gents, because we're about to take a long ride on the Magic Pod Bus to learn about DNA, specifically the structure, replication, transcription, and translation. I'll get through this as fast as possible to save both of us time. Right, now to start, we gotta explain how DNA is structured. DNA is built from nucleotides, which consist of a phosphate group, deoxyribonucleic sugar, and a nitrogenous base, which connect together to form long strands. Kinda looks like a twisted ladder, but of course that's also not quite right, but whatever. Anyway, the phosphate group at the end of DNA is called the 5' end, and the sugar end is called the 3' end. These strands are anti-parallel, which means that one strand runs 5 to 3, and the other runs 3 to 5, and they twist around each other to form a double helix shape. In between the backbones of these strands, there are nitrogenous bases, which make up the actual information of DNA. The bases are adenine, thymine, guanine, and cytosine. These bases can be sorted into two groups, purines and pyrimidines, which is based on its shape. Adenine and guanine are purines, the larger of the two, while thymine and cytosine are pyrimidines. Now, an important thing to know is Chargaff's rule. Who Chargaff is doesn't really matter, but what he said was, Chargaff's rule is that adenine and thymine can bind together, and guanine and cytosine can bind together, and nothing else. There's a small exception, which we'll get to later, but the bases, once paired, form hydrogen bonds in order to maintain the connection. Alright, now that's everything you need to know about the DNA structure. Now on to how genetic code gets cloned. Obviously, we need a method to replicate DNA, or we'd run out very quickly with all the cells dividing in our bodies. To begin, we gotta start somewhere, so our cells got a couple of points to begin called the origins of replication, where we begin splitting the DNA. The enzyme responsible for this first step of the process is helicase, who unwinds the DNA parental strands. When that happens, a replication fork forms at the points where the DNA transitions from together to being split. Single-strand binding proteins bind to the strands to stabilize them while they're apart. There's a joke to be made here, but let's ignore that. Then primase comes and attaches the primer to the open strand, which is where the new strands of DNA are forming. DNA polymerase then comes along and synthesizes and attaches new nucleotides to the primer and parental strand. How to know which nucleotide to add? Well, since there's only one option for what it can bond to each nucleotide, the polymerase reads the parent and picks the complementary piece. Not too terribly complicated, is it? Now, once the new strand is in place, ligase comes in and solidifies the connection, and DNA pol 1 replaces the primers from earlier. Also, while this is happening, topoisomerase is going to break and untwist the DNA to remove unnecessary strain. Sounds simple enough, right? Well, sorry to break it to you, but it gets more complicated than that. Remember from earlier how the DNA strands went in opposite directions? Well, that's going to complicate the process of creating the new DNA. Nucleotides can only be added to the 3' end of the DNA. If the DNA is added to that end, it's fast and simple, but if it's added to the 5' end, we have to slow down and do it in chunks. Because the 3' end is faster, the strand is called the leading strand, and the other strand is called the lagging strand because it's slow. On the lagging strand, the DNA is added in chunks called Okazaki fragments. Why are they called Okazaki fragments? Because Mr. Okazaki decided to, instead of naming it logically, he named it after himself like a prick. Getting back on track basically works like this. A primer is added slightly before the part that's being replaced, and then they work backwards till they hit another primer. They work their way up the DNA until they reach the replication fork. Does that make sense? Well, too bad. It gets even more complicated. Biology, man, you'd think it'd be more like mitochondria is the powerhouse of the cell. In addition, from the origin of replication, the DNA will be assembled in the direction of the nearest replication force. The DNA will be built in two directions, one quickly as the leading strand and one slowly as the lagging strand, which means depending on the side you focus on, the leading and lagging strand will change. Why do we do it like this? Hell if I know, that's just how bacteria do it, and we never bothered to update the program. Alright. Now that we've learned how to duplicate it, how do we use DNA? Well, our DNA is used to build proteins that our cells need in order to do work. To translate DNA into the proteins, we need to take the DNA to the ribosomes, but that's unsafe. We gotta make fake DNA called RNA to go to the ribosomes. Transcription is a similar process to replication in that DNA is split and a new strand is built from it. The strand which is used to build the RNA is called the template strand, which is the same strand every time the same gene is read. To start the process of building the RNA, we gotta get the RNA polymerase to bind to the promoter. To do this, there are transcription factors that initially bind to the promoter and then help in bring the polymerase. Then RNA polymerase binds to the ta-ta box, which marks the beginning of the promoter, and then moves down the gene until it reaches the terminal code where it continues a little further before detaching. As the polymerase moves down the DNA, it breaks apart and then connects to the corresponding nucleotide to form an RNA strand, then reconnects the DNA and continues down the chain. Congratulations, the primary transcript is completed. Too bad it's not going to be long before we come in and wreck it. Remember how earlier I said there was a small exception to the bonding rule? Well, it applies here. RNA is made with uracil rather than thymine. Since it replaces thymine, uracil bonds to adenine. Why does it do this? I couldn't tell you. Anyway, to begin the wreckage of our pristine primary transcript, we're going to have to add on to it. The RNA needs some protective bits to save it from the hydrolytic enzymes and such before it's sent out. On the 5' end, the RNA gets a 5' cap, and on the 3' end, it gets a poly A tail, which is basically just 6 adenines on the end. Now, this piece of RNA is going to be used to send information to the ribosome. This is called mRNA, M obviously standing for messenger. And with that, but wait, there's more. I completely neglected to talk about RNA splicing. On RNA, there are sections which are called exons, which are the important bits because they are expressed, and introns, which are removed because they're in the way. Why do we do this? Well, by changing which bits we keep and remove, we can produce a lot of different proteins from a single strand of RNA. This process is called alternative RNA splicing, which saves us a lot of space in DNA and time when we have to replicate the DNA. At least we did one thing that makes sense. Well, now that we figured out how to make the RNA, how does it help us make proteins? To answer that, we now finally get into the process of translation. Once the mRNA leaves the nucleus, it's going to bind to the small ribosomal subunit with the help of transfer RNA, or tRNA, which is going to be carrying the starting amino acid. Then the large ribosomal subunit will join the party and we can get this show on the road. By the way, ribosomes are made of specialized RNA called ribosomal RNA. Now, you may be asking yourself, how does a ribosome know which amino acid to add when building this chain? Well, the answer is simple. It doesn't. The tRNA I mentioned earlier actually does work because they're specially designed to pick up one amino acid and bind to one part of the mRNA. The part they bind to is called a codon, which is a group of 3 nucleotides, which is also referred to as triplet code. And then the tRNA will have a complementary set called the anticodon. Sorry to interrupt this riveting lecture, but the producers want me to share some fun facts. Number 1, there's only 64 possible oral triplets that can be produced. Number 2, 3 of the triplets are stop signs. 1 is start sign. The rest of them are amino acids. Number 3, the redundancies in our code, so different codon can produce the same amino acids, but there's no ambiguity, so one codon can't produce more than one amino acid. I hope you're still with me because it only gets worse from here. So to build the polypeptide chains, we got to go through the entire mRNA strand and get each of the codon amino acids added. To do that, we're going to feed the mRNA through the ribosome like a ticket muncher from Chuck E. Cheese. In the ribosome, there are 3 sites where the codon sits. The A site, which is where the cued amino acid sits, the P site, which has the long chain hosted to it, and the E site where the RNA is ejected. When a codon is in the A site, a tRNA binds to it with amino acid. Then the structure moves to the P site. Whatever amino acids were attached to the previous tRNA attached to the end of the incoming amino. This cycle repeats for the next codon with the now empty tRNA ejected from the ribosome until the mRNA runs out of codons. Once the mRNA has fully passed through, the newly completed polypeptide is released. Unfortunately, this is the end because the polypeptide chains aren't quite ready to rumble yet. They got to get in shape first. Sometimes they'll do it on their own, but sometimes they need outside help from other enzymes to fold correctly. Now, a small side note that's good to know is that there are 2 kinds of ribosomes, free and bound. Free ribosomes are chilling in the cytosol and generally make proteins for the cytosol function, while bound ribosomes are attached to the endoplasmic reticulum, and they make proteins that get shipped out of the country. In order to get the RNA to the right ribosome, ER-bound RNA is marked with signal peptide. This mark connects a signal recognition particle to the signal peptide that brings it to the ER. And with that, we've pretty much covered everything you need to know about DNA, minus other important things like mutations and chemical structures and the shady history behind the discovery of the structure of DNA and experiments done to find all this stuff out. But I'm not being paid to write a feature-length film, and I don't care enough to do so out of the goodness of my heart, and most importantly, I don't think it's required. Bye.